Hi, I'm Owen Traubert, and today I'm going to be talking about progress towards understanding the relationship between how the brain removes beta-amyloid plaque proteins and Alzheimer's disease. Alzheimer's. It's the sixth leading cause of death in the United States, a massive financial and care burden, and cannot be cured, prevented, or even slowed. Now, if we're talking about Alzheimer's, we have to talk about beta-amyloid. It's a protein found in the brain normally, but in especially high amounts in Alzheimer's. And when produced excessively, like in Alzheimer's, it accumulates in plaques. On the left is a normal brain, and on the right is an Alzheimer's brain. All of these red spots are accumulations of beta-amyloid plaques, which is a hallmark of Alzheimer's. These plaques result in neurotoxic effects, neural metabolic dysfunction, and inflammation, and they're a potential target for treatment. So our protein of interest is ABCG4. It's a transporter present in the brain, and it's been demonstrated to transport beta-amyloid in samples of the blood-brain barrier. It's thought to export beta-amyloid from the brain and into the blood. With this in mind, ABCG4 should reduce the presence of beta-amyloid in the brain, and therefore counteract the formation of beta-amyloid plaques. And because this plaque buildup is such a hallmark of Alzheimer's, ABCG4 should combat the development of Alzheimer's behavior. So our hypothesis was that Alzheimer's model mice with the ABCG4 gene knocked out should develop worse Alzheimer's-like symptoms sooner than Alzheimer's model mice with the ABCG4 gene functional. And other ABCG4 knockout mice without Alzheimer's genes expressed may have Alzheimer's-like symptoms on their own. So our mouse model is a line called J9, which contains both the Swedish and Indiana mutations, which cause human familial Alzheimer's. The Swedish mutation increases the amount of beta-amyloid, and the Indiana mutation increases the concentration of a more toxic subtype of beta-amyloid. We analyzed 4 genotypes: mice expressing ABCG4 and Alzheimer's genes, mice not expressing ABCG4 but expressing Alzheimer's genes, which we hypothesized would have the worst Alzheimer's-like behavior, mice expressing one copy of ABCG4 and Alzheimer's genes, and mice expressing no ABCG4 or Alzheimer's genes. We analyzed each sex separately and had 2 experimental rounds, one from 24 to 29 weeks and one from 62 to 68 weeks. We used 3 different behavioral tests: the open field test, which is a measure of anxiety, so more anxious mice should spend more time in the periphery versus the center; novel object recognition, which is a measure of conceptual memory, so ABCG4 knockout mice should spend less time investigating the novel object versus the familiar object; and novel object placement, which is a measure of spatial memory, so ABCG4 knockout mice should spend less time investigating the moved object versus the unmoved object. We found no difference in anxiety as measured by the open field test. For any of the 4 genotypes for either sex at either time point, there was no significant difference in the percent of time spent in the center. We also found no difference in conceptual memory as measured by novel object recognition in any of the 4 genotypes for either sex at either time point. And finally, we found no difference in spatial memory as measured by novel object placement for any of the genotypes for either sex at either time point, no significant difference. So because we saw no significant difference in Alzheimer's-like behavior between mice producing ABCG4 and mice not producing ABCG4, there must not be a crucial, unique role for ABCG4 in Alzheimer's. So either the lack of an effect can be explained by other proteins that transport beta-amyloid compensating for the absence of ABCG4, or possibly beta-amyloid transport across the blood-brain barrier isn't relevant to Alzheimer's, and other mechanisms can remove beta-amyloid and prevent or break down beta-amyloid plaques. So the implications from this are that ABCG4 shouldn't be investigated as a drug target, because if it is relevant to Alzheimer's, it's redundant. And additionally, other studies might analyze the effect of knocking out more types of beta-amyloid transporters to investigate whether or not beta-amyloid transport at the blood-brain barrier is even relevant to Alzheimer's. Here are my references. I've been Owen Traubert. Thank you.